Welcome to our presentation on sterility assurance, concepts and principles. Today, we'll explore the essential topics in the preparation of sterile materials, including aseptic processing, terminal sterilization, facilities, equipment, personnel, and quality control measures. Sterility is defined as the absence of viable microorganisms. However, practical limitations exist, as items labeled as sterile may not be completely free of microorganisms due to testing constraints. Sterility assurance is achieved through interrelated controls that ensure microbiological safety. The key elements of sterility assurance include controls, process design, aseptic processing, and terminal sterilization. Controls provide confidence, process design balances sterility and product quality, aseptic processing is used for products that can't be terminally sterilized, and terminal sterilization is preferred when possible. The production of sterile products is influenced by numerous factors that impact the sterility of the final product. These factors must be considered to ensure the highest level of sterility, even though not all influencing elements are depicted here. Key principles for sterile manufacturing include contamination control, personnel impact, automation, and separation. Contamination control focuses on preventing microbial ingress. Personnel are recognized as significant contamination risks. Automation reduces personnel interventions, and separation employs technologies to minimize human-sourced contamination. Quality by design, QBD, in sterile manufacturing involves unique applications, statistical challenges, design flexibility, and a primary goal of optimizing contamination control. QBD differs from typical pharmaceutical processes and focuses on microbial risk from personnel. This diagram outlines the key elements that contribute to sterility assurance. Each component plays a crucial role in ensuring the sterility of the final product, as described in the remaining sections of this presentation. Aseptic processing involves preventing the introduction of viable microorganisms into sterilized materials during assembly. Key principles include protecting materials from contamination and maintaining ISO 5 conditions when materials are exposed. The complexity of aseptic processing can vary significantly. Terminal sterilization is the preferred method for sterile pharmaceutical products due to its low risk. The process subjects the final product to sterilization with quantifiable safety levels, ensuring a probability of non-sterility, PNS, of less than 1 in a million. Post-aseptic processing terminal sterilization offers superior control over pre-sterilization bioburden and reduced lethality in the sterilization process. It extends the use for sensitive products and deactivates contaminants effectively, focusing on killing bioburden organisms. Containers and closures are integral components of sterile products, providing protection throughout the shelf life. Key characteristics include readiness for sterilization and minimal interaction with the product. Selection criteria and regulatory considerations are also crucial. Decontamination processes reduce microbial populations without necessarily achieving total kill. These processes include sporicidal and non-sporicidal treatments and are used for bioburden reduction of materials, equipment, and environments. Decontamination is not a substitute for sterilization. Common decontamination applications include controlled environments, material transfer, personnel, and equipment. Decontamination of clean rooms, RABs, and non-product contact surfaces, as well as periodic decontamination of operator gloves and product contact surfaces, are essential practices. Depyrogenation processes minimize pyrogenic contamination of surfaces, materials, and products. It is an essential requirement for injectable products and is used in various ways during sterile product manufacturing. Detailed guidelines are provided in USP chapter. Key considerations for equipment in sterile manufacturing include reliable operation, consistent product quality, and minimal adverse impact. Design features should ensure easy cleaning and sterilization, while flexibility and product protection are also important factors. Equipment in sterile manufacturing can be categorized into direct contact, indirect contact, and other equipment. Direct contact equipment interacts with the product, while indirect contact equipment impacts product quality without direct contact. Other equipment influences the manufacturing environment. Facilities design considerations include support functions, material flow, environmental systems, and cleaning considerations. These factors ensure the orderly transition of materials, components, personnel, and waste, while minimizing microbial, chemical, and particulate contamination. Classified environments operate in conformance with ISO 14644 series standards. Risk mitigation involves minimizing contamination from gowned operators, and pressure cascades control the flow of materials based on bioburden levels. Processing environments vary based on technology and background environment. Examples include conventional clean rooms, closed RABs, open isolators, and blow-fill-seal systems, each with specific ISO classifications and controlled air supply requirements. Conventional cleanrooms perform critical activities in ISO 5 environments with unidirectional airflow. Supporting areas are surrounded by ISO 5 to 7 environments for gowned personnel, and decontamination is commonly performed manually by personnel. Restricted Access Barrier Systems, or RABs, are designed to maintain a controlled environment for critical processes. They feature ISO 5 unidirectional air within the barrier, situated in ISO 5 to 7 cleanrooms. RABs are designed to prevent contamination through overspill and can be decontaminated manually or through automated systems. Isolators provide complete separation between personnel and the ISO 5 processing environment. They maintain a pressure differential between the ISO 5 environment and surrounding areas, ensuring contamination is minimized. Automated systems are commonly used for decontamination. Blow-fill-seal and form-fill-seal technologies form, fill, and seal flexible walled containers in ISO 5 environments. These systems are installed for various background environments and perform critical activities in unidirectional airflow. Closed systems ensure complete separation of production materials from personnel and the environment. They are designed to avoid human contact with sterile materials and can be sterilized either in situ or prior to use. Terminal sterilization environments can be accomplished in ISO 7 or better environments. Critical activities are performed within unidirectional airflow, and cleanrooms are commonly decontaminated by personnel. Products undergo terminal sterilization after filling. Active pharmaceutical ingredients require careful consideration of bacterial, endotoxin, and particulate contamination. Testing requirements are often specified in relevant USP/NF monographs, and microbial testing includes total aerobic bacteria, yeast, and mold counts. Excipients and process aids contribute to the microbiological quality attributes of the product. Specifications must ensure consistent conformance to critical quality attributes, and control measures include supplier controls and handling. Risk assessment considers origin and potential for microbial growth. Monitoring is essential to qualitatively assess the effectiveness of design and operational controls. It provides a snapshot of environmental conditions and highlights inherent constraints in sample size, location, and recovery capability. Excessive sampling can impair product safety. Viable monitoring methods include active air sampling, passive air sampling, surface sampling, and personnel monitoring. These methods detect culturable microorganisms in the air, collect airborne microorganisms, and assess microbial contamination from operators. Non-viable monitoring measures the number and size of particulates in the air. It is performed under static or dynamic conditions and uses calibrated particle counters for accurate assessment. Media fills and aseptic process simulations evaluate the performance of aseptic activity using sterile growth media. They verify the effectiveness of aseptic techniques and contamination control measures, representing processing conditions and activities. Sterility testing is a harmonized compendial test for sterile product release. It is required for release where parametric release is not approved and cannot prove sterility of all materials tested. Personnel in sterile manufacturing perform key activities such as cleaning, equipment operation, and decontamination. They are a significant source of microbial contamination, and control measures are essential to minimize exposure to human microbial contaminants. Personnel requirements include knowledge of microbiology, sterilization, aseptic processing, and contamination control. They must adhere to standard operating procedures, be proficient in aseptic techniques, and undergo microbiological monitoring after aseptic operations. Procedures in sterile manufacturing define operations determined effective through validation and experience. They are critical for ensuring the sterility of products and should be periodically reviewed for effectiveness. Interventions in aseptic processing include inherent interventions performed for every batch and corrective interventions for problems not performed for every batch. Examples include stopper jams, broken glass, and mechanical failures requiring manual correction. Intervention procedure guidelines emphasize risk awareness, process improvement, and designing for minimal risk. Sterilized tools should be used whenever possible to minimize contamination. Sterilization is the process that kills or removes viable microorganisms. It is the most effective means for microbial population control and is used for products, components, and equipment in sterile manufacturing. Utilities in sterile manufacturing include critical utilities like water for injection and process utilities like clean steam and compressed air. Quality requirements must meet USP monograph specifications, and system control ensures consistent state over time. Utility system maintenance practices include regular calibration, preventive maintenance, managed process changes, and ongoing process control. These practices prevent system failures and ensure consistent performance. Sterility is an unqualified concept, meaning the complete absence of viable microorganisms. However, demonstrating absolute sterility is challenging as there are no ready means to prove it. Testing for sterility has its limitations, as it only infers the sterility of a sample, not the whole. Therefore, the focus should be on process controls rather than testing. Establishing sterility assurance relies heavily on process controls rather than monitoring or sampling. Confidence in terminal sterilization comes from the process itself, not just testing. Aseptic processing involves a comprehensive approach, considering design, equipment, materials, procedures, and personnel proficiency. A holistic approach is essential, taking into account all supportive elements of operations. Several key references guide sterility assurance. Regulatory guidance from the FDA provides standards for sterile drug products by aseptic processing. Industry standards, such as PDA technical reports, offer insights into process simulation and aseptic processing. Facility design is guided by ISP baseline guides, and quality systems are informed by ICH Q8 on pharmaceutical development. Future trends in sterile manufacturing include increased use of automation and advanced systems to reduce human interventions. Closed systems are being developed for more advanced processing. Real-time monitoring technologies are being implemented for continuous oversight. Single-use technologies are expanding, offering more disposable systems in sterile manufacturing. Sterility assurance faces several challenges. The increasing complexity of biologics and combination products adds to the difficulty. Regulatory expectations are evolving and can be stringent. Integrating new technologies with established practices requires balance. Effective risk management strategies are essential for maintaining sterility assurance. Quality by design in sterility assurance involves a thorough understanding of product characteristics and the manufacturing process. It includes assessing the design space, which is the combination of process parameters. Risk assessment is crucial to evaluate potential failure modes and their impact on sterility. Developing a control strategy ensures a comprehensive approach to consistent sterility. Continuous improvement in sterile manufacturing is driven by ongoing data analysis and trending of environmental monitoring data. Regular technology assessments help identify new tools for implementation. Training programs are essential for continuous education and skill development. Process optimization is an iterative refinement based on accumulated knowledge. Global harmonization efforts include ICH guidelines for technical requirements for pharmaceuticals for human use. The PIC/S scheme provides standards for GMP. WHO standards offer guidelines for pharmaceutical manufacturing. Industry collaborations involve cross-industry working groups and conferences on sterility assurance. Emerging technologies in sterility assurance include rapid microbial methods for faster detection and identification of microorganisms. Isolator technology offers next-generation isolators with improved decontamination and monitoring capabilities. Process analytical technology enables real-time monitoring and control of critical process parameters. Artificial intelligence is being used for predictive maintenance and quality control. The future of sterility assurance involves a holistic approach, integrating design, controls, monitoring, and continuous improvement. Technological advancements are embraced while maintaining fundamental principles. Risk-based strategies focus on critical control points and science-based decision-making. Global collaboration continues to drive harmonization and shared best practices.